The modern biology from which this whole presentation and all this stuff is really garnered, and most many schools use this book. It's in chapter 9, and in this cheetah book, it's in chapter 12. So it really doesn't matter. All of the same information there presented a little bit differently. Hello, sir. Making homework for the science. Oh, right behind you.
Where's your circus pen?
Yeah, where's your pen? All right, Will, what we're doing is we're going to blast through Mendelian genetics, so you can take notes or not. I need to check the crossword, and then we'll have some time to work on it, okay? Remember, the crossword's due completely tomorrow, and we're going to have a quiz over it next week. Tomorrow we're going to do after we go over the crossword to make sure you got them all correct, which I'm sure you will, if they wouldn't fit. Basically, we're going to do a kind of a cool little activity, a fun activity, kind of applying this stuff, kind of a good way of ending this little mini short week. So, with that said, let's go on. Okay, so Mendelian genetics, that should be the title, obviously, of your notes. The reason for that is because it's named after this guy whose name you need to know. If you do, don't write it down. I want you to know it was like the 1800s. You don't need to know these dates specifically, but you should know this happened long ago. Okay? And what I really want you to know, I don't really care if you know he's Austrian, but he developed the laws of inheritance, and he is known as a father of genetics. I think that's the next, hang on, I'll take the next screen, it says the same thing. Yeah, father of genetics right here. And because he's the guy who came up with all these laws and the things that we're going to go through today of inheritance, right? So you need to know that. If you do know that, don't write it down. I don't care if you know Johann, if you know Gregor Mendel, I'm okay with that. Okay? And the cool part of the, I mentioned yesterday, he had huge sample sizes, right? We're good? All right. Yeah, okay, copy that down. Just kidding. Okay, that's what this guard looked like. So just okay, and you don't need to write this down, but the key here is that what I want you to know is, when we talked about this yesterday, is Mendel didn't know anything about chromosomes, right? The technology didn't exist at that point. We didn't really know, barely could see cells at that point. But yet, all the stuff we're going to talk about pertains to what happens there. So basically, that's what I want you to know, okay? I don't care if you know any of the particles or whatever, but just make sure you know. He didn't know anything about DNA. It was pretty significant. All right, so terms we went over yesterday. These are three terms that you probably already know because you probably did these in your crossword last night. But you need to know trait, heredity, and genetics. And self-explanatory up there, right? Basically, study heredity, the traits, anything you pass down. Now, traits are things that are like your color, hair color, and all that kind of thing. But also, there's some things you don't see, right? I mean, your heart chambers, whether you have a heart or not, liver, all that kind of stuff. That's all traits too. It's basically anything that can be coded for on a gene that can be translated and transcribed into a protein, basically. Okay. See, somebody got your crosswords or had them as we go. That's good. We good? Yes. You guys got to kind of, again, remember, you got to kind of keep me in check from going too fast and stuff, all right? All right, so when we do crosses, and we're going to be doing a lot of these, obviously, remember, there's two types. There's a monohybrid, single, mono 1, die two crosses, right? And basically they are what they are. Monohybrid cross, one trait, pick a trait, whatever this happens to be, flower color. And dihybrids two, and that's really what you need to know. Those are two terms, again, that you probably have to use in your crossword. Monohybrid, single cross, dihybrid, two-factor cross. Now, are there more than two factors? Of course. I mean, when you consider like a human being, how many different traits are there? I mean, gosh, I don't even know, thousands. And when you study them, yeah, you can go there. The problem becomes is that it becomes astronomically complex, and therefore it's beyond the scope of us. Okay, so you're going to try hybrid crosses, which I did one, and I'll never do another one. It just becomes incredibly laborious, and it's not necessary because once you get the concepts between these two, you really don't need to go beyond a dihybrid cross. It illustrates both this law of segregation and the circle which we're going to talk about. Okay. All right, Punnett square, you guys know this. This is that little square that kind of helps you do this. Kind of look at what the possible outcomes are going to be of parents. Parents go here. Possible kids are here, right? You take the parent phenotypes up here. Male, in this case, female here, doesn't really matter. It's kind of arbitrary where you put them, but, whatever. And the combinations that come about are the potential offspring. That's what goes in here, and the parents go out here, if you remember that, right? All right, and you're going to basically get crosses, and you can get like a story problem, and you probably remember, and the terminology here is what you're doing your crossword for: heterozygous, homozygous, dominant, recessiveness. All these terms are required to really do these problems, okay? And that's why we're hitting that hard up front. So in this case, just as an example, we'll come back to this in a second, basically. If you have here's the example problem. Basically, you have two heterozygous parents. You cross. They're yellow peas, and you need to know that yellow is dominant in this case. And dominants is represented as capital letters, if you recall. Recessive are the lowercase letters. So if you had a cross like this, you just distribute them, male, female, whichever, and then you run the crosses, you pair these two up, and you get a Punnett square. Everybody good with this, right? It's like, 8th grade stuff, basically. And then when you look at what they actually look like, by the way, is the glare too bad here, guys? Can you see that okay? Or anybody? Good? All right. It's like the eye test. Good? Better. Good? All right, so here we go. So basically, and you look at the results, and then you basically come up with some very powerful predictions, basically ratios. And that's really the beauty of this Punnett squared combined with the knowledge that basically Mendel provided us. And then ratios are basically the relationship of this to that to this. So a genotypic ratio is the types of genes and how many they are in comparison to the other types of genes. So in this case, you get a 1 big Y to 2 big Y little y's to 1 little y little y. That's your genotypic ratio. In this case, is what do you see? You see 3 yellow, you see 1 green. Your phenotypic is what they look like, is 3 to 1. Now, I blasted through that very quickly because we really didn't introduce these terms. So we're going to go through the terms, and we'll come back to that, okay? First off, dominance, recessiveness. This is, probably boring, but dominance means it's going to be seen if it's there. It's dominant. Convention says that's capital letter. Okay? Whatever the trait. Now, what letter you use is kind of arbitrary. Usually it's like the first letter of the color, like red is big R, blue is big B, whatever. Okay? You remember that? That's arbitrary, but the capital part isn't. If you put a capital down, you're basically saying that's a dominant trait. And how do you know that? Well, you either are given it in the problem or you reason it out based on the results. Okay? Recessive is just the opposite. Recessive never shows up unless it's by itself. Dominant traits always dominate over recessive traits. Little case little. Nothing new here, probably, right? Ma'am.
For the ratios back at the previous slide, how do you know what order to do it? Like, wouldn't it work as 2, 1, 1, or like?
Yeah, we'll get to that. That was kind of introduce it. We're going to go through each of those, and we'll come back to that.
Okay.
So, yeah, it's usually dominant, heterozygous, or hybrid, and then recessive. That's typically the order you do it in, and,
Okay.
and we'll get to that. Okay, are we good here? Everybody, okay? So, oops, wait a second. Come on, baby. There we go. Okay, alleles. Let me make sure I hit this one, because this is a term that oftentimes people get confused on the difference between a gene and an allele. Okay? And let me just give you the way that I remember. A gene is just a physical piece of basically of the chromosome. Okay? And again, Mendel didn't know that, but we now do. You can actually take and splice genes, and that's I think we kind of talked about that as one of the applications and the reasons we want to understand genetics is because genes are a physical piece that you can take, remove from even one species, put in another, and have them do their work, like insulin, for example, in bacteria. Okay, so genes are physical parts of the DNA or chromosome. The alleles is the different expressions for that gene. So when they get transcribed, how can they be translated and transcribed into? For example, color. Could be red, could be white, could be pink, whatever. Those are the alleles, the adjective, the descriptive words of this gene. Okay? So that's the expression. Most of the time, and you can imagine there are different types of expressions, and you may remember some of those from when you did this before, but most of the time there's two. Okay? The dominant and recessive. But many times there's blendings, and we'll go through the different types. There's codominance and some of the terms that you may have come across as you went through your crossword last night. So there's many different ways that genes are expressed, but the most common type is this dominant recessiveness. In fact, we're going to get to this. One of the laws that Mendel provided us is a law of dominant recessiveness, which basically says that genes are expressed this way. Okay? Get that? So again, allele is the different forms of a gene. A gene is a physical, tangible, concrete part that you can touch. Okay? All right, getting to those words. Types, genotype, phenotype. Genotype, again, is the types of gene combinations there are. Big R, little r, little r in this case. Okay. And the phenotype is what are they going to look like? The adjectives, again, that you see, the descriptor words. Okay? And for some reason, people kind of get these confused too. And I don't really understand why. But, again, for me, the gene types, the pheno is just what they look like. All right, good? Am I going too fast? Too slow, probably. All right. So here's an example of an equation that kind of puts it all together. If you got a flower that's red, this is kind of really nicely done because you have a big R and it's red. Okay, dominant trait. Yellow would be the recessive trait, little r. And you go ahead and you pair them. Question for you: why do these letters appear in pairs? Okay, but why? Why is there one of each? Why is there two alleles? Yes?
It's like when one's from the mom, one's from the dad.
One's from the mom, one's from the dad. On what? You're right. One what? Chromosome. So these genes, remember, we humans have 46 chromosomes, 23 pairs. Those pairs are homologous pairs. Old terminology we learned before, right? But now it comes into play in a kind of a practical way. So moms and dads. So mom or whoever has the big R, dad has the little r. during sexual reproduction, the two come together